Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the living organisms. And so far, what we have discussed about the classifications, evolutions. We have discussed about the different types of biomolecules, their structure and functions. And then in the previous module, we have also discussed about the central dogma of life or the molecular biology. And then in the previous lecture, we were discussing about the some of the cellular processes, and in that context, we have discussed about the immune responses and how the antibodies can be generated within the host and how they are actually imparting the different types of immune responses. So now in today's lecture, we are going to discuss about the some more cellular processes. But before getting into the detail of the cellular processes, we have to understand the techniques which are actually going to be used for the understanding the different types of cellular processes. So as you know that the cells are could be of different types. So cells which are present in the blood, right? So cells can be present in the blood. It can be present in a complex sample like the liver. It could be present in brain, and it could be also present in heart. So you see in the blood we have the RBCs, WBCs, lymphocytes, neutrophils, basophils, eosinophils, platelets. Similarly in the livers you have the hepatocytes, Kupffer cells, blood. Then you also in the brain you have the neurons, astrocytes, glia cells. Then in the heart you have the cardiomyocytes, cardiac pacemaker cells, fibroblasts, smooth muscle cells, and the endothelial cells. Now all these cells are varying in the different types of properties, right? What are the different properties? Actually, these cells are varying. They are varying in terms of the density of the cell lysate, right? So every cell has the presence of the different types of organelles, different types of biomolecules, and that's why that constitute for the density of that particular cell. Then the cells can vary even in the size of their shape, right? So they can also vary size, like for they can be as small as the 10 micrometer, or they can be as big as the 30 to 40 micrometer. Then the cells can also vary in terms of the metabolic reactions, right? And then the cells can also vary in terms of the receptors what are present on their cell surface. If you see the size, right, what you see here is I have given you an example of different types of cells like the sperm cells, red blood cells, lymphocytes, and what you see here is they are varying somewhere around 3 micrometer to as big as the 40 micrometer. So they are very big and they are very different, right? So if you want to analyze these cells and if you try to understand some of the basic metabolic or the cellular processes, you require a machine which actually can be able to perform these particular tasks. So if you want to prepare this kind of machine, what you require is you have cells of different size. You can also have the cells of different densities. That's why I've given you the different colors, right? Now what you require is you require a fluidic system, so that these cells are going to be present individually, and then when these individual cells are going to be illuminated by a laser beam, it can be detected by the different types of detectors. So you can have a detector which can actually measure the size. You can also have the detector which actually can measure the density. Then you can also detect the different types of fluorescent signals, right? And ultimately, if based on this, if you require, you can also collect these cells for further analysis. So this particular type of instrumentation is being called as the cell sorter or the cell analyzers. Okay, and a typical cell analyzers is going to have all these components. You can have the fluidic components, you can have the illuminations or the excitation component, and then you can also have the acquisition components. As far as the instrumentation is concerned, you are going to have a laser beam, right? So you can first going to have a fluidic system where a stream of fluid is going to run, right? And that fluidic system is going to give the single cell suspension, and once the single cell suspension is going to transfer through the nozzle, The single cell is going to be illuminated by the laser beam. So these laser beam, then the cell is actually going to give the signal, right? So it can actually give the signal to the different types of detectors. So it can actually give the signal to the PMTs, and that is actually going to be give you the signal about the forward scattering. Then it also can give you the signal to the another PMTs, which is actually going to give you the signal about the side scattering. And then it also can give you the signal for the different types of fluorescent signal, like the you can have the FL2, you can have the FL3, and you can also have the signal for FL1. So all these signals are going to be collected by the ADC system, and that is actually going to be analyzed by a computer. So that all that analysis can be done, and that's how the flow cytometry, or the instrument which is going to be used for the analyzing the different types of cells, has the three main component. One, you're going to have the flow system, which is called as the fluidics. Then it's going to have the optical system, which is actually going to use for illuminating the objects with the laser beam and then collecting the different types of signals. And then you're going to have the electronic system, which is actually going to use for the signal processing. uh, in the fluidic system, you're going to have the flow cells, and these flow cells are actually going to streamline the cells, so that it is actually going to going to give the individual cells for the optical system. So it's once the cells are present in the single cell, then these single cells are going to be illuminated by the laser beam. So then in the optical system, you're going to have the laser beam, right? So you're going to have the light source, which actually going to be used for the exciting the laser, the cells, right? So you can have the different types of optical system. You can have the laser beams, or you can also have the arc lamps. So both of these sources can be used for illuminating the objects. And then in some of the latest flow cytometer system, you can also have the organ laser, which actually can excite the samples. Then we have the optical system. So optical system, you can have the different types of detectors. You can have the forward scattering channel, you can have the side scattering channels, and the forward scattering channel, the intensity of this signal has been attributed to the cell size and the refractive index, right? So the forward scattering is going to give you the important information about the size of the cell, whereas the side scattering is actually going to give you the density of that particular cell, right? So then we can have the electronic system. So the electronic system, the job of the electronic system is that it is actually going to acquire the signal from the optical system, and then it is actually going to process. So the electronic system is going to convert the optical system into the photoelectrons, and then it actually going to measure the amplitude, area, and the width of the photoelectron pulse, and it amplifies the pulse either linearly or logarithmically, and then digitizing the simplified pulse. By utilizing the electronic system, you can actually be able to plot the data in the three different formats. What you can do is you can actually plot the single color histograms, you can color two color dot plots, you can color two color contour plot, or you can make the two color density plots. Okay. Histogram plot, like the single color histogram plot, is something like this, right? So it's going to give you a histogram, right? So you can have the single color like the FL1, right? So you can have the fluorescent signal, you can have the number of events on this side, and it is actually going to give you a histogram. It's going to give you a peak like this. Okay, so this is called as the histogram plot, whereas it can also give the two color contour plot. So in that case, you can have the, like, for example, side scattering plot, or the forward scattering plot, right? And then it actually can give you a contour plot like this. So it's going to give you the dot 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 like this, right? So that is a contour plot and two color plot. And then it can also give you the contour plot. So this is the contour plot, two color contour plot, right? So it can give you a contour of the signal, right? And then it can also give you the two color density plot as well. There are multiple steps in which you can actually have to do the data acquisition. So first you have to bring the sample into the your buffer, right? So mostly the people are using the buffer like the PBS or any buffer which is osmomolar to the cells. So you're going to have two different types of cells, right? You're going to have the unstained cells, right? And you're going to have the stained cells for whatever the reason, right? You can. So with the unstained cells you are actually going to optimize the parameters, right? You're going to optimize the different types of parameters like what will be the voltage, what will be the gain, and all those kind of parameters, and that's how you are going to actually ensure that the peak what you are going to see is actually in the middle of the this particular graph, right? So it should be at the level of 10 to power 1, so that if the peak will go either in the backward direction or in the forward direction, you should be able to monitor the movement. Then you can just keep these settings constant, and then you can start collecting the stain samples. Apart from this, once you collect the sample, you might be actually getting a lot of data, right? You might be getting dots, right? So in some cases, you might be only interested in looking at some of the subpopulation within the sample, right? So that can be done by a process which is called as the gating, right? So with the gating, you can be able to collect some of the data. So gating, as the name suggests, you can select only a certain population of the cell for analysis on a plot. It allows the ability to look at the parameters specific to only that subset. You can have the different types of gates. You can have the rectangular gate, so you can have the rectangular gate. You can have the elliptical gate, you can have the polygonal gates, you can have the quadrant gates, and you can also have the histogram gate. So the dye is what you can also use in the flow cytometers. You can actually use the FITC, phycoerythrin, you can use the PERCP, you can use the APC, you can use the Texas Red, and so on. And all these are actually having the different types of wavelengths. So according to wavelength, you can use the either FL1 or FL2 or FL3. So these all this can be used according to the intensity. Now, what are the different types of biological processes? So, if you see a cell is actually going to, and if you provide the nutrition, right? If you provide the media, right? Remember that when we are talking about the how to can actually be able to prepare the media, it will actually going to grow for the growth, right? So once it will grow for the growth, it is actually going to increase its number. So that the first biological process is that how you can actually be able to monitor the increase in number, the growth in terms of increase in number. Apart from that, when the cell will actually going to, if there will be a loss of nutrition, then the cell is actually going to undergo under the stress. So if there will be minus media, right? For example, if you stop feeding the cells with the fresh media, then they will be under the stress, and under the stress they can actually go through have the different pathways, right? They can also go with the pathway of autophagy, and they can also go with the and the autophagy is a cell survival pathway. So autophagy will allow the cells to grow for some more time and going to stay for some more time. But if that time is also going to be over and the stress is going to be even beyond that, then the cell has two choices. Either it will go for the apoptosis or it can go for the necrosis, and both of these events are eventually going to lead to the death of the cells. So you can have the increase in number. So that is the first event. The second is you can also study how the cells are going through the apoptosis or the necrosis or the death pathways. Okay. So when we talk about the cell proliferations, the cell proliferation can be understood like this, that you have a cell, right, and it has a nucleus, right? So and I'm sure you might have, so this cell is surrounded by the plasma membrane, right? And this plasma membrane is, remember that when we were talking about this plasma membrane, it is selectively permeable, right? So if it is a selectively permeable, it will not allow the entry of the molecule spontaneously. So it will not allow the entry of the molecule as per the concentration gradient, especially like the charged particles, either negatively charged or positively charged, will not going to pass the plasma membrane, because the plasma membrane is made up of the lipid or the protein molecules, and that's why the plasma membrane will not allow the charged particles. Charged particle has to be go with a facilitated transport, and this is what is going to happen. So when you are going to provide the nutrition, right, or you're going to provide the medium to the cell, it will actually going to start dividing the nucleus, right? So it's going to have the growth in terms of nucleus, right? And then there will be a cell division, right? So then there will be a cell division, and that's why the single cell is going to be get converted into two different cells. But you can imagine that if there will be a loss of nutrition, right, so then the cell will instead of going like this, right, so if there will be a minus nutrition, the cell will actually go for the death pathway, and once it will enter into the death pathway, it is actually going to form the holes onto its plasma membrane. So it's going to have the plasma membrane selectively permeability is going to compromise, and if that will going to happen, it will going to start taking up the charged particles from the which are present in the environment. So this is one of the way in which the people are studying of the cell proliferation, and they can actually be able to use that for counting the different types of cells. What you can do is you are actually going to mix the cells. So you take the cell suspension where you are actually be interested to see how many cells are live or dead, and then what you can do is you can just add the small amount of trypan blue. So trypan blue is actually a charged dye. So and as I said, in the here example, right, if the cell is live, so this is a live cell and this is a dead cell, right? So if a live cell will have a plasma membrane integrity maintained, then it will not allow the charged particles to go inside, and as a result this cell is not going to take up the stain. Whereas in the case of the dead cell, because the plasma membrane is lost its selectively permeability and it's lost its ability to stop the entry of the charged particle, the charged particle will enter, and as a result it is actually going to be it will take up that molecule. So because of that, if you stain the cell suspension with the trypan blue, which is a charged dye, it is actually going to give you the cells with the appearance like this. So this is the cells without any dye, and this is the cell which is actually having a dye. So this is actually a dead cell and this is a live cell, right? So that you can actually put into a hemocytometer. So hemocytometer is having a chambers onto this, and these chambers you can actually be able to visualize under the inverted microscope, and then you can be able to count these cells. So trypan blue is a charged dye, and viable cell exclude this dye to the presence of plasma membrane potential, whereas the dead cells in the presence of absence of plasma membrane potential accumulates the dye in the cytosol. So what you are going to do, you are going to take the glass slides, cover slips, you require the hemocytometer, and then you also require the hemocytometer and inverted microscope. You remove the cells from the cell plate by trypsinization or the EDTA treatment. Then you plate a small amount of cells onto the cover slide and cover them with the cover slip. You mix the 50 microliter of cell suspension with the 50 microliter of trypan blue, and you fill that into the hemocytometer chamber. Observe the cells under the 20x objective using inverted microscope with the face plate, and the viable cells are actually going to appear colorless, whereas the dead cells are actually going to appear as the blue or the dark colored. So the viable cells are actually going to appear as the colorless, right, and the dead cells are actually going to be appeared as that they will take up the dye. So this is going to appear as the blue color. And there's a discrete procedures through which you can actually have to load the hemocytometer with the stained cells and all that. So that we have discussed in this small demo, where the student has discussed how you can actually be able to stain the hemocytometer, how you can be able to stain the cells with the trypan blue, how you can load the hemocytometer with the into the cell. and then how you can be able to count under the inverted microscopes.
First, we have to remove the remaining media, then trypsinate the cells, then we will count the cells and see it. After cells are detached, we have taken into clean falcon, then we have to centrifuge the cells. As these cells are very delicate, we have to centrifuge at 1500 rpm for 2 minutes. Now we have to remove the supernatant and resuspend the cells in fresh media. After resuspension, we have to count the cells. So I am going to take 20 microliter of this cell suspension and mix with the 20 microliter of trypan blue and count under Newbar chamber. Before counting, we have to see how a counting chamber or hemocytometer look like. This is a typical hemocytometer, also called as Newbar chamber, which contains these squares in upper side and lower side, with each square having depth of 0.1 mm and area of 0.0025 millimeter square. Now I am going to put a cover slip on this chamber, then I will add slowly cell suspension. Through capillary action, it will spread all over the squares. So we checked how many cells are there in all the squares. Now we have to count. How to count the cells? So here a typical Newbar chamber which contains squares, 5 squares. So we have to count cells in these squares. So each square is an area of 0.0025 millimeter square and total small squares 16. So total area of this whole square is 0.04 millimeter square. So the depth of this each well is 0.1 millimeter. So what is the volume? 0.04 into 0.1. So that is total 0.004 millimeter cube or 0.004 microliter. So say we have combined cells in each well. Say this is A, B, C, D. Here we have 100. Here we have 150. Here we have 110. Here we have 100 again. So the total cells we have to take average. That means 100 plus 150 plus 110 plus 100 divided by 4. Total 4 squares we are counting. The average is 115. So 115 cells in 0.004 microliter volume. So how many cells per 1 ml? So that we can calculate simply 0.004 into 1000. That will give the volume cells per ml.
Now, next, let's move on to the next biological event. So, this biological event is where we can also be able to study the cell cycle. So remember that we have already discussed that the cell will go through with the different phases within the cell cycle, where you can have the G1 phase, the S phase, G2 phase, and the M phase. Now what you have to remember is that the amount of DNA is going to vary among these phases, right? So amount of DNA is going to be one copy of DNA in the case of the G1 phase, and the two copy of DNA is going to be in the S phase, right? And the 2 copy will continue as the 2 copy in the case of the G2 phase, and as well as in the M phase it is going to be divided, right? So you see that the DNA content is varying when you are going from the G1 to S and G2 and M phase, and at this stage there will be a cell division, and because of that there will be a sister cell is going to be formed, and the main cell is again going through with the cell cycle. So what you can do is you can take the cell and you can stain it with the DNA dye, right? So if you take the stain it with the DNA dye, it is actually going to the DNA dye is actually going to bind the nucleus, bind the DNA within the nucleus, and that's why it is actually going to give you the staining for the DNA, and that can be used as a measure. So what you can do is you're going to have the two different types of cells. You can have the cells which are unstained, right? And you can also have the cells which are stained with the DNA, right? So DNA dye. For example, you can use the propodium iodide or some other dye. So stained with DNA dye. And what you can do is you can just analyze these two cells under the flow cytometry, and that is actually going to distinguish between the amount of signal what is coming from the different types of cells, and that's how you can be able to study the cell cycle events. So this is what you're going to happen when you see the data, right? So data of this cell cycle is like going to be like this. It's going to give you this. So this is the G1 phase. So you see the amount of DNA is less, right? And then this is going to be called, so as you see, right, it's going to give you a pattern like this. So it's going to give you a pattern like this, then it goes like this and that. So this is actually the G2, this is the S, okay. So because you see the different phases, okay, it's going to give you the proportion of the cells under the G1 phase, G2 phase, and the S phase, and you can be able to use that to calculate, or you can be able to use that to study if there are the conditions which are compromising the cell cycle events, or if there are compounds which you are trying to develop so that they will interfere with the cell cycle. So that is also we have prepared a very small demo to show you how you can be able to stain the cells, how you can be able to process the samples, and how you can be able to analyze the data.
For checking the data, we have kept it on the setup so that we can see whether the data is coming properly or not, and then we can remove it from the setup and then acquire the data. So now we are going to load the sample onto the sample injection port and then press acquire. As we can see that the number of events have started recording. In the FSC and the SSC plot, we can see that most of the population is seen between the 200 and the 200 mark. The population which is coming away from the is that of the doublets. In FL2A and SSC, we can see that there are three different populations, G1, S, and G2. And in the FL2A and the counts, in which you can see that the longest peak is the G1, the between one is the S, and the smallest peak is the G2. So before acquiring the data, we have to set the number of events. So we go to acquisition and storage and press record 10,000 events. So in order to record the data, we have to remove it from the setup and then setup and then press acquire. In the FL2 and the FL2W channel, we can see that there are two different populations. One is a thin line and other is the presence of some doublet or clumps of cells. This distinct population might be presence of the doublet cells or clumps of cells. So while acquiring the data, we can see this, but in the FCS Express software, we can remove that. So now that we have acquired the data, now we can take the data from the treated sample. We have to remember that we don't have to change the parameter conditions in order to check the compare the untreated and the treated samples. Now we have to change the sample name from untreated to treated, and also change the file count to 1. It's okay, and then acquire. We can see that there is a little shift in the S and the G2 phase, but we can't say for sure that there is a change in the data. So for that we have to acquire, we have to process the data on the FCS Express software in order to see what is the difference between the untreated and the treated sample. So now we'll remove it from the setup and then acquire the data for the treated sample. Sometimes we can see that the events per second will be low, so in order to increase the events per second, we can pause the recording and then tap the sample once or twice in a while to shake the sample, and then the flow will be continuous again from the sample injection port, and the events per second will increase. Now we can see that the events per second has increased because we have tapped the sample, and the data acquisition will be a little bit faster. So now we have acquired the data for the untreated and the treated samples. In order to see in which cell cycle phase of the cell cycle the arrest has taken place, so we have to process the data in the FCS Express 5 software. We have to now process the data, so for that purpose we use the new layout. As we can see that there are multiple options available: dot, density, color, dot, contour, surface, histogram, and multi-cycle DNA and kinetics. For this cell cycle DNA, we only need multi-cycle DNA plot and the dot plot, so we are going to open these two. Because we have recorded our data on the FL2A channel, and also because the propydium iodide is only shown in the FL2A channel, we are going to open the FL2A channel. So we have opened the dot plot and the cell cycle DNA plot. We can see the how much population of cells is represented by the G1, S and G2 by right clicking on the plot and then selecting statistics and then show DNA cycle statistics. So a small window will appear in which it will show that the percentage G1, the percentage G2 and the percentage S. So this is for the untreated sample. Let's see for the treated sample. As we can see that there is some change in the untreated and the treated sample, both in the both plots, the dot plot and as well as the cell cycle plot. So in order to see how much percentage of the cell cycle phases have changed, we right click on the plot, then show DNA cycle statistics, and then place it right beside the untreated one in order to compare. So we can see that there has been a reduction in the G1 phase from 61 to 49, and from G2 also from 15 to 12, but there has been an increase in the percentage S phase, which has increased from 23 to 38. So we can say that there is a significant change in the phases of the cell cycle, but we can only be sure after doing the experiment in the triplicate, so that we get the proper standard deviation and also the standard error. So this is one way of processing the data.
Now, what we have discussed about the growth, right? And we have discussed about how you can be able to monitor the increase in number. But under the stress conditions, the cells has the two options. One is the cell survival pathway, which is also called as the autophagy. So that is going to be a survival pathway. And what is mean by the autophagy is that autophagy is eating yourself. Okay, auto means self and phagy means eating. So if you eat yourself, uh, it is called as autophagy, which means under the autophagy conditions, the cell is going to start utilizing the cellular material for its survival. Okay, which means it is going to start recycling the cellular material. For example, you can have the different amount of mitochondria, you can have the different amount of chloroplast, you can have the different amounts of other organelles. So what you can do is, and cell probably, say for example, if you have 500 copies of the mitochondria, right? So if you have a 500 copies of mitochondria, what you can do is, you can, or what the cell is going to do is, it can actually digest the for example 200 copies. Okay, so if we digest the 200 copies, so if it digest the 200 copies, and if we digest utilizing the lysosomal enzyme, then it is actually going to produce two things. It is going to produce the the biomolecules, right, and it's also going to produce the energy. And both of these things, the biomolecule as well as the energy, is going to utilize by the cell to run its basic metabolic reactions, or it's going to utilize that to pass the time, because these cells are under stress. They are not getting any kind of nutrition, so they have to wait until the nutrition is going to appear. So during that phase they can start utilizing the cellular content. For example, they start eating up the different types of organelles, different types of biological biomolecules, and so on, right? So that they can be able to produce the biomolecule which are essential for the survival, and they can also be able to produce the energy. And that all these events are coming under the process which is called as the autophagy. Once the autophagy events are going to be failed to provide the sufficient energy and sufficient biomolecules, then the cell will actually going to go for the death pathway. Okay, and within the death pathway you can have the two pathway. One is called as the apoptotic pathway, the other one is called as the necrotic pathway. And within the apoptotic pathway you have the two different types of pathway. So that we are going to discuss when we are, and how you can be able to study the apoptosis as well. So apoptosis is going to utilize the, so apoptosis is going to utilize the, so you can imagine a cell, right? So cell can have the is happening because you have the too much stress, right? So if you have too much stress, it is going to induce the apoptosis. But this stress could be external stress, right, or it could be internal stress, right? So okay, now for example, if you add some drug molecules, right, this is going to be an external stress. Okay, so these external stress are actually going to be received by the receptors what are present onto the cell surface, whereas the internal stress, there could be an internal stress also, like for example there will be a lack of nutrition, the production of ROS, that all those kind of things are going to be the internal stress. So because of that, it is actually going to activate the two different pathway. One is called as the extrinsic pathway, so it's going to be called as extrinsic pathway, and the other one is called as the intrinsic pathway. uh, Irrespective of the there are different events what is going to happen when you are going to activate the cell surface receptors, and then it is actually going to activate the extrinsic pathway, or when it is actually going to activate the extrinsic receptor. Ultimately both of these pathways are actually going to activate the caspases. Okay, and these caspases are eventually going to activate so caspases are proteases, right? So these caspases are proteases, so they will actually going to activate, they are going to start degrading the proteins. Okay, and the caspases are eventually going to activate the DNases. Okay, and these DNases are then actually going to start damaging the DNA. Okay, and so there will be a damage of proteins, there will be a damage of DNA, and because of that the cell will eventually going to go for the apoptosis. Then what will happen, the cell is actually going to engulf the material, and it is actually going to trap these degraded material into uh, the membrane bound structures, and these membrane bound structures are called as the apoptosome, right? And these apoptosome are then going to be released, okay, into the blood or into the tissue, and then they are actually going to be taken up by the other cells for the clearance. So they are actually going to be cleared from the circulation. So that's why the apoptosis is considered as the safe mode of death, because it is actually going to it is going to release the apoptosome, and these apoptosomes are going to be cleared from the circulation without getting the too much inflammations and other kinds of immune responses. Now, if you want to study the apoptosis, what you can do is you can actually be able to first study what will be the DNA, right? How, whether the DNA is damaged or intact. So you can actually be able to study the DNA damages, or you can also be able to study the alterations, what is going to happen onto the plasma membrane, right? So one of the major alteration what is happening onto the plasma membrane is that it is actually going to have the reversal of the lipids composition. So mostly the phosphatidylserine, which is actually lipids, is always be present onto the inner surface of these plasma membrane. But under the apoptotic phase, it is actually going to flip, and then it is going to be present onto the external surface of the plasma membrane. So that also can be detected. So there will be a lipid symmetry which is actually going to be disturbed during the apoptosis, and that can be detected with the help of the different types of analytical tools, or you can actually be able to study whether the DNA is intact or the damaged. DNA can be studied by the different types of DNA binding dye. For example, you can use the propadium iodide or the PI, right? And phosphatidylserine can be studied by a probe which is called as the annexin 5. Okay, so if you stain the cells with the and then you can do a checkerboard analysis. So you can do a checkerboard analysis and in the facts. So what you can do is you can stain the cells with the propylene iodide and annexin 5, and it is actually going to give you a pattern like this, where the inner quadrant or the first quadrant is actually going to give you the cells which are healthy, okay, and they are going to be, so these, if you do a checkerboard analysis, all these cells are going to be considered as the healthy cells, okay. Now what you see here is this is the control cells, okay, or the untreated cells, whereas these are actually the treated cells, okay. So when you treat the cells with some agents which is actually going to activate the extrinsic apoptotic pathway, what will happen is you see that the number of cells under the first quadrant is going to be reduced, and the cells are now moving into the all other quadrant, right? So it is, they are moving into the quadrant number 2, 3, and they are moving into the quadrant number 4 also. This quadrant number 2 is actually having a high signal for FL1, but they are actually having the lower signal for FL3. So these cells are called as early apoptotic cells, okay, because they are actually having the very low signal of the FL1. FL1 is going to be give you a signal about the PS, and FL3 is actually going to give you the signal about the PI. So they are actually having the DNA damage, but the DNA is not going to have the very high quantity or high signal for the PI, okay. And then you can have the this quadrant. So in this quadrant you have the high propadium iodide signal and the high annexin. So this is actually a late apoptosis, okay. So because it is actually showing the PS and as well as the DNA signal, whereas in this one you have the low PS signal and the high DNA damage signal. So that is actually a dead cells or the necrotic cells, okay. So this is the way you can actually be able to identify the different types of species of the cells what is present in the into the apoptosis. How you can do that? You can actually take the cells and you can be able to stain it with the DNA dye, right? And you can also use and stain it with the annexin 5, okay. So when you do that, okay, it is actually going to stain the cells, and then you can actually be able to study or analyze these cells under the flow cytometry, okay. and so flow cytometry, and then you can do a checkerboard analysis, right? So when you do the checkerboard analysis, it is going to give you the cells of the different species. Apart from this, you can also use the another method where we can also use the two different combination of the DNA dye, like the propadium iodide, and we can use the acridine orange. Okay, and that is also a very common or the popular method of detecting the apoptosis. So we have prepared a small demo, and it is actually going to give you a clear idea how you can be able to study the apoptosis within the cells.
So after adding the acridine orange and propadium iodide to the cells, we have to acquire the data on the CellQuest Pro software. So the first thing we do is open the CellQuest Pro and connect it to cytometer. And then we need the counters, the detector and amps, and the status. For the acridine orange propadium iodide staining, we need two dot plots. One is for the FSC, SSC for the forward scattering and the side scattering, and the other one is for the FL1 and FL3. The FL1 plot is on the x-axis, whereas the FL3 plot is on the y-axis. The FL1 plot is for the acridine orange and FL3 plot is for the propadium iodide. After taking the plots, we have to set the directory and save the data in our required location. In the detector and amps, we have to remember that we have to set the population of the healthy cells in the first quadrant, that is 10 to the power of 1 and 10 to the power of 1. So after we set the untreated cells in the first quadrant, and then we analyze the treated cells, and then we can say whether there is any shift in the fluorescence in the untreated and the treated cells. For the treated cells in the third and the fourth quadrant, that represents the apoptotic and the necrotic cells. So now we will be taking the sample, but before analyzing the data, we have to set the number of events to the 5000, and then keep it on setup, and first we will see whether the events are coming properly or not. Now we press acquire. As we can see that in the FSC and SSC plot, we can see the events coming near 00, that represents the healthy population, as well as in the FL1 and FL3 we have set the healthy population between the 10 to the power of 1 and 10 to the power of 1. So this represents the first quadrant. We will show in detail how to do the quadrant analysis in the FCX Express 5 software. Now that we have set the population in the first quadrant, we will remove it from setup and acquire the data. After the untreated samples, we have to take the treated samples on the same parameter description which we have set for the untreated cells. Now we change the sample in the sample injection port to the treated sample. We have to remember that we don't have to change the parameters, or else we will never be able to say whether there is any shift in the untreated or the treated cells if we change the parameters. After changing the sample, now we have to choose the directory for the treated cells, and then change the name also to treated, and also the file count to 1. Then press OK, and then now we acquire the data on the same parameters. As we can see that there is some shift in the population of the cells. The population is having a little bit more fluorescence than the untreated cells, which represents the apoptotic and necrotic cells in the third and the fourth quadrant. In the fourth quadrant, mostly the necrotic and the dead cells are present, whereas in the third quadrant the late apoptotic cells are present. After we take the untreated and the treated samples, after we acquire the data for the untreated and the treated samples, we have to analyze the data in the FCS5 Express Pro software using quadrant analysis. In the quadrant analysis, we can see how much populations of cells are present in which quadrant, and therefore we can identify the number of healthy populations and the apoptotic and the necrotic cells. So after acquiring the data in the FACS equipment, we have to now analyze the data in the FCS5 Express software. So the first thing we do is we open the new layout and change the orientation to landscape, and then now we input the data. First thing we do is we take the untreated file and then open the dot plot. We need two dot plots. The one is the FSC SSC, and the other one is the FL1 FL3. The FL1 FL3 dot plot shows the live and dead cell staining. The FL1 is responsible for the acridine orange, whereas FL3 is responsible for the propydium iodide. Now we take the treated file and again we select the dot plot. In dot plot as well, we need the FSC and the FL1 FL3 plot. As we can see that there is difference between the untreated and the treated sample. Now we have to find out how much percentage of the cells have gone actually the apoptotic or necrotic. Like we have to divide the population of cells into 4 quadrants using the quadrants option. So we go to the gating and then take the quadrant and then apply it on the FL1 FL3 plot. We have to apply it in such a way that we cover all the cells in the untreated plot. So let's say that in the untreated plot we are having 92% live cells and 7% are in the early apoptotic phase. And then after applying the quadrant to the untreated, we have to apply the same quadrant to the treated one in order to find out the difference between the two. So just we click on the quadrant and then we copy and paste on the treated one. So in this way we can say that in the first quadrant in the untreated sample we have approximately 93% of live cells, whereas in the treated one we have like in the treated one we have only 35.7% healthy, whereas the 33% have gone are in the late apoptotic and 29% are necrotic cells. So in this way we can use acridine orange and propydium iodide to determine the healthy, the apoptotic and the necrotic cells in different treatments. And also we can establish a relation between different concentrations of treatment as well and the number of live and dead cells in any experiment. So this is the way we analyze and process the data on the FACS equipment in order to do the live and dead cell staining. Hopefully this video was helpful.
So I'm sure you might have understood the different processes, what is happening with when you want to study the apoptosis of the cells utilizing the flow cytometry. And with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more aspects of the cellular processes. And we have discussed so far. We have discussed about the basics of the flow cytometry and how this technique can be used to study the cells of the different sizes as well as the different densities. And this is a very robust technique what we can use for studying the different types of cellular processes. And then utilizing the flow cytometry, we have studied about the cell cycle, we discussed about the cell proliferations, and then ultimately we have also discussed about the apoptosis. So with this, I would like to conclude my lecture here. Thank you.